I have a question.
Sure, go ahead. Yeah.
So in quiz one, the last four or five, yeah, last four question was 5 points, right?
It depends. Everybody get a little bit different quiz.
Um,
So I'm not sure what your last four questions are,
Uh,
but yeah, but some questions are worth more than others. Yeah, but I can take a look at yours
yeah, because
later on.
unfortunately I didn't pay attention to time, and in last four question my time was out of, you know,
Oh,
I
right,
couldn't
right,
do
right.
it. I'm so sad, but
Yeah, okay. Yeah, I can take a look at your quiz and then, I'll send you a message after that, okay?
okay.
Yeah.
Okay, thank you.
All right. Good afternoon, everyone. First of all, I think you guys did quite well as a class for the online for the quiz number one. Class average is 79, I think, which is quite high. Okay. Generally speaking, average is usually in the low 70s or on a good day, it might be like a mid 70s. I haven't get a high 70s for some time. So, well done. I hope you continue to succeed in the course. And if you didn't do as well as you hoped to, hopefully after the first quiz, you have a better understanding as to what to expect. And if you do need extra help, we have the tutors available, and myself is available. As well if you need some clarification on some harder concepts and whatnot, feel free to reach out. Anyone have any comments about the quiz or any, concerns, suggestions before we, continue with the lesson? So I heard a time might be an issue for some of you. Just
Yeah.
Can
yeah.
I ask
Sure, please
one?
go ahead. Yeah.
Which is the highest mark of this?
Oh, so I think some people got perfect. Yeah. So that is quite good. Yeah. Mhm.
Okay.
Yep. So pace yourself, right? Especially with those short answer questions. You don't want to, go into you don't want to spend too much time on them. Okay. Just answer the question in a concise, clear manner, and you would get the full points for them. Okay.
Okay.
So let's quickly review some of the organelles that we've learned last class, and then we're gonna move on to lecture
Oh,
four.
sorry, teacher.
Y-
When our grade will be published?
I already published the grades. Are you not able to see it? Did you guys? Can you guys not see it?
I see it. I see it.
Oh
Oh,
yeah. I
sorry, I
Oh,
don't
that's
know.
okay. Yeah, no problem. Oh, I just remember something. Because it's an online course. So we do not return the quiz for you, it's for you to, go through the questions because we need to keep the test bank, maintain its integrity, right? We don't want it to, go out and be out there on the internet, right? So for that reason, we do not return the quizzes. I know it's not ideal situation, but that's one of the constraints of online learning. Okay, so I hope you understand that. All right, here we go.
Sorry, I just have a
Sure,
question,
yes.
Vincent.
Yeah, yeah.
When is in the course is going to show like grades? I know it has like the average, but it doesn't show like A or B or C or like the
Oh,
other courses.
I mean, I can post information about that, but basically anything above 80 is an A. Anything
Okay.
above 90 is A plus. Okay.
Okay.
Above 70 is a B. Okay. Yeah, that's
Okay.
basically the range. Yeah, I can find like a table or something, and then I'll post it as an announcement. If not today, tomorrow. Okay.
Yeah, because some
Okay, thank
courses
you.
have that grade letters, but
Yeah.
course is not, unfortunately.
No, yeah, no, I mean, I could switch it to letter, but I think it's more beneficial if you know your actual percentage, because
Hmm.
that will allow you to calculate, later on what you need in the test in order to achieve certain final grades, right? Because if I tell you you get an A, 89 is an A, and 81 is also an A, right? But those are very different in terms of getting a high average and low average, right? You know what I mean? Yeah.
I mean, we can see both of them, A and also you know,
In other courses,
we can see, I mean, in chemistry, yeah, we
you
can
can see
see
both.
grade. Yeah, we see both.
Oh, okay. I'll take a look. Like I said, it's a new system, so maybe I just have to, enable some kind of option there. Okay, I'll take a look. But for the most part, you guys can see your grades, right?
Teacher, can we
Mm-hmm.
get any feedback from you about quiz?
Like I said, right, like I cannot show you which question you get wrong, but like if you're looking for specific feedback, then maybe you can send me an email and then, I can take a look at your quiz and give you some general idea, okay? Yeah. All right. Let's take up this like I said, like I'm not particularly concerned, right? Looking in the class grades, generally it looks quite good, okay? So here, you have this picture in your study guide. Hopefully you printed it out. And we have the cell membrane here. Okay. We have a pair of these things which are found in animal cells, but not plant cells. We call them centrioles. And if you forget what centrioles are, they produce spindle fibers that help guide chromosomes during cell division. Okay. Over here we have another structure. Anybody want to tell me what that might be?
Ribosome?
Uh, okay, these little dots would be ribosomes, but what about this bigger guy right here? Anybody? It's part of the and
Paroxysome.
yeah, okay, yeah. So it could be a peroxisome or it could be a lysosome. It's hard to tell. So I'm not gonna ask you to label that on the test, but this particular one, it's a lysosome. Okay. Sorry, I gotta restart the program. Gets a little bit weird when you use it for too long. Okay, right here. Okay, so that is the lysosome. Lysosome helps digest macromolecules, okay, or ingested particles that are taken in by the cell. They can also trigger apoptosis, which is programmed cell death, right? Other things, we have this guy. I'm sure you remember that. That is the mitochondria. And what does the mitochondria do? Anybody?
Produce energy.
That's right. Yeah, energy production. Okay. These little small dots here and there, like this guy, right? Oh, it's hard to point to it. There we go. That's the ribosome, and they are involved in protein synthesis. Some ribosomes are floating freely. Others, they are going to be attached to another structure. Right? So these things over there, maybe I should switch the color. These things with the ribosomes on them, you can see the dots. That would be the rough endoplasmic reticulum. So on the next quiz, there might be some questions where you have to key in the names of these things. Always type the full name, mind your spelling. Uppercase, lowercase doesn't matter, okay? But like you gotta spell it right in order to get the mark. This would be protein production, protein synthesis with ribosome plus packaging. Okay. Over there, further out, it is the smooth endoplasmic reticulum. Can someone tell me the function of this thing? What does it produce?
Lipid.
Yeah, that's right. Lipid synthesis. Okay, it also
Create hormones. Okay.
that's right. So lipid includes hormone, right? Very good. Also break down lipids and toxins. How about that? What do we call that?
Body.
That's right. That is a Golgi body. Golgi body, which is responsible for sorting, sorts, modified, and repackage. Repackages protein. Okay. So the rough ER, the smooth ER, the Golgi body, along with the lysosome, right, and the nuclear envelope actually, they are all part of the endomembrane system, right? There you go. So these little lines that you see here throughout, that's part of the cytoskeleton. A couple other things. Nucleolus in the center, which makes ribosome. And this is where you have the chromatin. You can't see that color. Chromatin. There you go. Any questions before we move on? You can label the plant cell. There's an additional cell for you to look at. But any questions at all before we start today's lesson? Nope. All right. Well, today we're going to be learning about the cell membrane. And first thing we're going to do is we're going to draw the cell membrane. So please take out a piece of paper and, I suggest putting it sideways. And then we're going to be drawing like here, 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 here. And then we'll write some notes here, here. Okay, so plan your space accordingly. All right. So our cell membrane is something called the fluid mosaic model. Fluid mosaic model of the cell membrane. Of cell membrane. So what is mosaic? Let me Google a mosaic window for you. Okay, so this is a mosaic window, right? You see with it's like it has multiple, oops, I don't know. It has multiple parts to it. It's not just one thing, right? There's like little green triangles here, purple triangle, whatever, right? Like different components coming together to form the entire window. So that's what the cell membrane is. It's not just made up of one thing, it's made up of multiple things together with different components, and they all play an important role. And the other description is that it's fluid. It's not like a wall that's behind me, okay? If it's a wall, then it's solid, nothing goes through, right? Very rigid. We don't want that. Right? We want the cell membrane to be fluid to allow material to move in and out of the cell. And the fact that it's fluid means it is not a static structure. Okay, it constantly can shift, right? Things on the cell membrane can shift places, and it's very dynamic. Okay, so what we're gonna do is we're gonna take the cell membrane. This is the cell. This is the nucleus. We're gonna take a slice of the cell membrane, and we're gonna make it really big. Okay, there you go. So the cell membrane is made up of primarily phospholipids. We learned that in the second lecture. So I'm going to draw a bunch of phospholipids here, and you can draw along. Okay, some of them will have the saturated fatty acids, and every now and then you can draw anywhere you like. Some of them will have unsaturated fatty acid. Okay, like this. Okay, so there you go. Leave a gap. I'll tell you what's in the gap later. Okay, and then leave another gap. Okay, so draw that. And we're going to draw the other half of the cell membrane as well. This is just one side of it. Let me trim it a little bit. There we go. So it looks more uniformed. And then on the other side, we're going to have this as well. Okay. And again, some of them will be saturated tails, others might be unsaturated. Okay, I'll tell you the meaning of that later on. There we go. There. So this whole thing is the cell membrane. Cell membrane. Okay, it's two layers of these phospholipids. So we say phospholipid bilayer. Phospholipid bilayer. Two layers of phospholipids. That's what it means. Now you might recall the phospholipid, if I draw it one more time, that it has a hydrophilic head and that it has hydrophobic tails. So in this phospholipid bilayer, you have the hydrophilic heads either facing the outside of the cell or the inside of the cell. The inside of the cell, we already know it is called the cytoplasm. It's, mostly water. That's why we have the hydrophilic heads facing it. And then the outside of the cell, I don't think I've told you the name yet. It is called ECF, okay, extracellular fluid. Okay, so that's outside and this is inside. Just gonna give you a moment to copy these things, and then we'll go on. Meanwhile, let me see if I can change the way the grades are being displayed. Okay, so let's take a look at what else is on this cell membrane. Okay. Well, actually, you know what? Let me give you another quick note here first. Okay. Phospholipids molecules can move freely within its layer, but they do not flip to the other layer. Okay. So what I mean is, if you take a look at this phospholipid can shift left, can shift right, left, right. It can go this way and this way, but it's not gonna flip and go to the other side. Okay? So, this one's not gonna flip and go to the other side. But they can move, front and back and left and right. I want you to understand this is just like a cross section of the membrane. The whole membrane, don't draw this, but the whole membrane is it's like 3D, right? So like you have more phospholipids here, more phospholipids. Do you understand? Right? It's like a whole sheet of these things, but that's too complicated to draw, which is why I'm only drawing like a cross section of it, right? I guess one of the ways to think about this is, hold on, I gotta show you a picture of what I mean. Like you guys know what a ball pit is, right? Like those kids playground thing, these ball pits. It's kind of like that for the phospholipid, right? Like these are the phospholipid heads, and the balls can shift front, back, left, right. Okay, they can travel around, and it's not going to be a problem. Right? But these things are not going to flip over to the other side. Is that okay? Now, why do I have these gaps here? Well, it turns out phospholipid is not the only thing that exists in the cell membrane. You also have proteins that would insert themselves in the cell membrane. Some proteins will go all the way through. Right? It's going to insert throughout the entire bilayer. Other proteins will just kind of attach on the side. Right? So if it's attaching on the side, then we call it a peripheral protein. Peripheral literally means on the side, like your peripheral vision, your side vision. We have another peripheral protein here. I hope you're using color for this. After last lesson with the cell, I hope you realize it's important to use colors, right, for the notes. Hopefully you've brought some colors with you to class today. So this one that goes all the way through, we call it an integral protein. Integral protein. Okay, the definition is in the PowerPoint, but I mean, the picture here should be pretty self-explanatory. Integral goes all the way through. Peripheral, just on the side, either outside or inside, it doesn't matter. Okay, so these proteins are important for various reasons. Okay, we're gonna look at 6 different functions associated with these proteins, but very briefly, they are there to serve as anchorage point to make the protein, to make the cell membrane structurally sound. Okay, without these proteins, if it's just made up of phospholipids, then the whole thing is just gonna fall apart. It's not strong enough, the lipids itself. By having a little bit of these proteins, it's like having a cornerstone, right? When you build a house, you need something to anchor all these things onto the membrane, and that's what the proteins are for. Right? If these proteins are destroyed, if they're denatured, for example, then the whole cell membrane is going to unravel and the cell is going to die. So I want you to keep that in mind. Now, in addition to proteins, we will also have some cholesterol, okay? Cholesterol here and there. Okay, so not a lot, but there are cholesterol. Okay? Cholesterol, the function is to modulate, to control, okay? The fluidity, the membrane fluidity. Okay? So basically, if the phospholipids pack together too closely, we learned in lecture 2 that it's going to turn it into a more solid state, right? And the cholesterol are basically there to keep them apart. Right? They prevent the phospholipids from getting too close to each other. And so if you have more cholesterol, then it means the membrane is going to be more fluid. On the other hand, if you have less cholesterol, then nothing is preventing the phospholipids from packing together, then it's going to be more solid. Okay, so depending on what kind of cell membrane we need, the cholesterol content is going to be different. Okay, similarly, the type of phospholipids you have will also affect the fluidity. Okay. You might remember saturated fatty acids tend to pack together closely. So if you have more saturated fatty acids, more saturated phospholipids, then your membrane will be more solid as well, right? Okay. Sometimes you will have proteins that have sugar attached onto it. So these are little glucose chain. Right? It's hard to draw hexagon, so I'm just not doing a very good job here. But that's a sugar chain. Right? This is carbohydrate. And if you have a protein that has carbohydrate on it, we call it a glycoprotein. Protein is not the only thing that can have sugar on them. You might have glycolipid as well. Okay, glycolipid. We'll talk more about later. One of the things these glycoprotein and glycolipids do is cell identity. Okay, it allows your cell to recognize each other. Right? All the cells in your body are gonna have similar glycoproteins and lipids that gives it a unique identity. So if something outside of the body invades you, your immune system would recognize them because they do not have the same glycoproteins and glycolipids that you have. Okay. Now, finally, on the inside of the cell, you might have some of these cytoskeleton. And the cytoskeletons, they might be actin, intermediate filaments, right? Which helps maintain the cell shape. And some of them are going to attach to the glycoproteins or the integral proteins, right? Or the peripheral proteins. And that's basically the fluid mosaic model, right? All these different components coming together to form the cell membrane. That's the mosaic aspect. And they're not locked in place. They can move around, okay? So that makes it fluid. Any questions about any of the stuff on the note before we go back to the PowerPoints? All right. So let's take a look at this question. How would the percentage of saturated, unsaturated fats, I guess I could change it to unsaturated, saturated phospholipid, okay, because that's the one that we're talking about in the cell membrane, and cholesterol content of a cell membrane found in plants growing in cold environment differ from those growing in hot environment. All right, so let's write this out, and hopefully it will make sense. Okay, so let's do a table to summarize these information. Okay, so we have hot environment, and then we have cold environment. Okay, and so what kind of adaptation do we need? What about percent of saturated phospholipid? Percent of unsaturated phospholipid? And percent of cholesterol. Okay, so write those down first, and I'll tell you about it. Okay, so if a plant grows in a hot environment, like, the desert or something like that, do you think it's better for the plant to have a more solid cell membrane or a more liquid cell membrane? What do you guys think? More solid or more liquid?
More liquid.
More liquid, okay. Anyone else? Liquid, and the person say liquid. Okay, let's do a poll and see what you guys all think. Okay. Let me repeat my question before you choose. If a plant lives in a hot environment, do you think the plant needs to have a solid cell membrane, or B, a more liquid cell membrane? Right? More solid or more liquid? Okay, just a couple of minutes. Locked in your answer. Okay, we are kind of half-half with the response. So think about it. If it's really hot, then things tends to melt, right? You put butter on the stove, it's gonna melt. So we don't want that. We don't want the cell membrane to melt, right? Therefore, to counteract the high temperature, the adaptation that the plant cell needs is more solid cell membrane, right? It's already hot, and if you make it more liquid, then it will melt even faster. Do you understand? Right? For people who chose more liquid, have I convinced you that the correct answer is more solid? Or it's still, nah, not too sure. Right? You don't want it to melt, so you want to make it more solid. So how do you make it more solid? Do I have more saturated phospholipids or less saturated phospholipids? Which one would make the cell membrane more solid? Which one would pack it even closer?
More.
More, that's right. So you need more saturated. Remember, saturated packs really close and it's going to be more solid, and you need less of this. And for cholesterol, also less. Remember, cholesterol is trying to keep the phospholipids apart from each other. So if you want them to be more solid, you don't want to keep them apart. So you need less cholesterol. On the other hand, if it's a really cold environment, then the adaptation would be a more liquid cell membrane. The cold temperature is going to make it more likely to freeze, right? So you don't want it to freeze. You want it to stay liquid. If it becomes frozen, then nothing goes through, right? Okay, so you need to keep it more liquid, and to make it more liquid, you will need to have less saturated fat, more unsaturated phospholipids, and more cholesterol. Okay, this is important. Please make sure you understand this before you write quiz 2. Got it?
So people who are living in cold country, they need more cholesterol?
Oh, good question. No, it doesn't work like that. This would have already happened. Like if you look at the cell membrane of plants that are growing in hot environment, this would be true. Okay, and then this would be true for cells for plants that are growing in cold environment. It's not like it would change. Like it's not like if you put a plant from hot environment and start growing in a cold environment, and then it would change the composition, right? It doesn't work like that. The plant's likely going to die, right? For the same reason, that's why people who live in cold or hot environment, their body's not going to shift the cell membrane composition, okay? We are very different from plants because we can maintain a core body temperature of 37 degrees Celsius, right? Okay, so our cell membrane doesn't have to adapt for it because it's always in a 37 degrees Celsius environment,
We ourselves
regardless of how
can
cold it is. Right?
we ourselves can control our body temperature, homeostasis.
Exactly.
Okay.
Very good. There you go. Okay, we're gonna move on to membrane function next, and membrane protein functions. Right? So all those proteins that we drew, they actually play important roles in various ways. So just to quickly recap, if the protein goes all the way through, then it is going to be an integral protein. If it just attach on either outside or inside, they are called peripheral protein. Okay, so here are the definitions you can see right there. So what do they do? Well, number one, they are useful for attaching to cytoskeleton. I already show you in the drawings, there are some cytoskeleton on the inside, but there are some on the outside as well. So in order for the cell to attach and interact with these cytoskeleton, you would need to have the membrane proteins. Okay, so this is a red blood cell, and red blood cell, as you will learn next semester, has a very distinct concave shape in the middle. Okay, so the cell membrane is actually indented in the center, but how do you keep it indented? What's anchoring the cell membrane at those locations? Well, it turns out it has to do with the membrane proteins being anchorage points for those cytoskeleton. Okay, here you can see all these lines. They are fibers. They are cytoskeletons on the outside of the cell. And for the cell to attach to these cytoskeleton, once again, you will need to have membrane proteins. Okay, next function is cell signaling. Okay, cell signaling. Some membrane proteins, they are receptors, and the receptors allows certain molecules to bind to it, and then it's going to cause some changes inside the cell. Right? So the receptor, since they're made out of protein, therefore the shape of them are very important. In this example, because of the shape over here, they would only be able to respond to these type of signaling molecules. If you have a signaling molecule that's like circle, then it would not be able to interact with the receptor. Okay, so each type of receptor will only be responding to a particular kind of trigger. Okay, so an example of that would be adrenaline. Okay, so adrenaline is released from your adrenal gland. It's on top of your kidneys, and you release adrenaline when you are, for example, excited, when you are scared, when you're in danger, right? Or like when you are like psyching yourself up, pumping yourself up to do a race, then you have adrenaline in your system. Think like, riding a roller coaster, you would also have adrenaline. Think driving on the highway, and then maybe you were going a little bit too fast, and then you see a cop car on the side, then the adrenaline start pumping, right? So these adrenalines is going to go into your bloodstream. The blood will take it throughout the entire body, but only cells with the correct receptor will respond to it. Okay? Remember, the shape of the receptor has to match the signaling molecule, right? So not everything will react to adrenaline in the body, even though they are all exposed to it when it's released from the adrenal gland. Okay? So once the adrenaline binds to it, adrenaline is also called epinephrine. Like, yeah, remember people like you might have heard of people who suffer from like severe allergies. What kind of pen do they need to help them? Do you guys know? Like peanut allergy or something? What kind of pen do they have to
EpiPen.
carry? What is it? I'm sorry?
EpiPen.
EpiPen, that's right. The epi is the epinephrine, okay? Which is the same word as adrenaline. They are two words for the same thing, okay? So when the adrenaline binds to the receptor, it will cause a series of reactions to take place inside the cell. We just call these the signal transduction pathway. The name is not important, right? But it's a series of reactions which ultimately will change how the cell behave. So in this case, the person is getting ready for a run. He did his warm up. The atmosphere causes him to build up the adrenaline, and then his cells will respond to the adrenaline and cause the glycogen to release glucose, right? Which makes sense. You're gonna do a race. You're gonna need a lot of energy, and you get the energy from burning glucose, right? So that's what the membrane protein does in this case. Okay, it's a receptor, and it relays the signal and causes the cell to change its behavior. The signaling molecules never went into the cell, okay? It just stayed outside, and then it's like pressing a button, and all these things happen inside the cell, okay? Quick story, which might help you remember this. Whenever I teach this concept, it reminds me of my childhood, okay? Uh, my parents, they didn't like the idea of Halloween, okay? Something about handing out free candies. They didn't like that idea, okay? So every year when it's Halloween, and someone pressed the doorbell, right? My mom would be like, high in the basement, turn off the lights, right? So that's what we're gonna do, right? We all rush to the basement until there is no one ringing the bell, and then we come back up, right? So that never really makes sense because, like, I tell my mom, yeah, they can see the light, they can see that you turn off the lights, right? But it's like this example, right? The protein and the membrane, it's like the doorbell. Okay, the kids who are ringing the doorbell, they never came into the house, but by ringing the bell, it causes so many changes that are happening inside the house, right? And that's kind of what the receptor protein does. Okay, it relates the signal and causes the cell to change its behavior. Okay, you will see this in neuron as well. When the brain cells communicate with each other, they also rely on a lot of these signaling molecules binding to receptors, and then it's like a chain reaction from one neuron, it hops to the next neuron. Don't worry about this too much. We have a whole, 2 weeks of lecture on this thing next semester. I'm sure you're looking forward to that. Okay, next function, next membrane protein function, right? It's enzymatic activity. We already know some proteins behave like an enzyme, so that's not surprising. And in this case, if you want to do a chemical reaction, the enzymes are actually all lined up in the cell membrane. Right? In many cases, the reaction is not complete after going through one enzyme. Right? So like you might put in the circle and it converts it into this molecule, but that's not the end molecule. You want to feed this into a second enzyme, and then you get another intermediate and so on and so forth. So this is actually 4 different reactions before you can get to the end product. And by having the enzymes all lined up right one next to the other on the cell membrane, you can allow the reactions to happen at a much quicker and more efficient manner. Okay? It's the same reason why we keep all the cooking stuff in the kitchen, right? When you use it, you take the chopping board and then the knife is right here, and then you have your cooking frying pans, or with the spatula. Everything is all in the cabinet, right? You can grab them easily because it's all in the same place. Rather than keeping things, maybe you keep your spatula in the kitchen, in the bathroom, or you keep the cooking pans in the basement. Then you have to go all over the place to get all these things when you need them, right? So that's exactly what happens here. We keep all these enzymes in the same place rather than having them randomly scattered in the cytoplasm. When we do need them, they are all right next to each other, and we can do our job much more efficiently and much quicker that way. All right. So 3 functions so far: attachment to cytoskeleton, cell signaling, enzymatic activity. Do you guys have any questions before I tell you the other 3? Okay. Next one is intercellular junction. We sort of mentioned this last class when we talked about intermediate filaments. There are places in the body where we want the cells to be tightly locked with each other. We don't want any gaps between them, right? In the intestine, for example, in the blood vessels, we don't want blood to be seeping through these cells. We don't want the contents of our intestines to be leaking out. And so it's important to stable them together. And that's what the membrane proteins can do sometimes. Okay, there is a membrane protein that locks this cell with this cell. Same thing over here. So even things coming along, they're not able to seep through it and leak through. Okay, so they create these things called intercellular junction. Number 5, some proteins and some lipids, they have sugar attached onto them. We drew it already in our picture earlier. We call them glycoproteins and glycolipids. Glyco just means sugar, right? So sugary proteins and sugary lipids. So collectively, they form an identification tag, and that allows our immune system to distinguish self versus non-self, right? When a bacteria invades your body, it does not have the same set of ID tags, and your white cells is going to be able to find them out and basically destroy them. Right? In this picture, this pink guy is a white cell, and this is an invading cell that does not share the same ID tag, and it's being attacked by the white cells. Okay, your ID tags are going to be different from my ID tags. No one in this world is going to share the exact same ID tag as you, okay? Unless you have an identical twin. Which is why when it comes to organ donation, you can't just like randomly take a kidney from someone and then plant it into someone else, right? Because of the difference in the ID tags, the body will reject it, right? Now, like I said, no one else has the exact match as you. So we match it as much as we can. And then people who receive organ transplant, they have to take medications to dampen their immune system, right? To minimize the chance of organ rejection. Okay, so that's cell to cell recognition. Okay? And part of this has to do with what kind of sugary proteins and sugary lipids you have on your cells. Is that clear? Last function. Okay, last function for membrane proteins that we're going to look at is transport. And that is the main topic of lecture 4. Okay, transport. Proteins in the membrane allows movement of typically molecules that are very big or molecules that are charged. They wouldn't be able to cross the cell membrane on their own, and therefore they rely on these membrane proteins to move them over. Okay, well, that's a lot of talking. I'm going to launch a poll question for you to try. Go ahead. Okay, anyone else? All right. Thank you for those of you who have participated. Let's take a look at this. According to the fluid mosaic model of a cell, we're looking for the true statement. Phospholipids, they are free to depart from the membrane and dissolve in the surrounding solution. No one chose that. That's good. This wouldn't be good, right? If the phospholipids can freely leave the cell membrane and dissolve, then the cell membrane would unravel on its own, right? Killing the cell. They occur in an uninterrupted bilayer with membrane proteins restricted to the surface of the membrane. That is not necessarily true. The membrane proteins doesn't have to be on the surface, right? Look, they could be on the inside. They could be going through the cell membrane, they are not necessarily just restricted to outside. So that's not true. They have hydrophilic tails in the interior of the membrane. That's not true. It's supposed to be hydrophobic, right? When we drew the phospholipid bilayer, we have all the tails facing the inside. The phospholipids can move laterally along the plane of the membrane. That's the correct one. Okay, I told you in the notes that these things, it's like the balls in the ball pits. They can move front, back, left, right, diagonal, right? What they cannot do is they cannot flip to the other side, right? So that's why the last one is not true. There's no flip-flopping, okay? Correct answer is D. Any question about that? Okay, let's do another one. All right, just a couple more seconds and I'm gonna close this. Okay, it's pretty straightforward. I hope this one. It's basically the 6 things we talked about, and you're looking for the ones that is not on the list, right? So protection from bursting, and that's not the function of cell membrane. Okay, very good. Let's take a quick break. When we return, we're gonna take some more notes. Let's come back at 1:35. All right, and here is the grading scheme that you guys were asking me about. You can take a picture of it if you want. I tried changing the percentage. I'm not sure why you would be able to see both the percent and the letter grades in chemistry. Maybe I'll talk to your chem teacher and find out. I'll see her tomorrow. Maybe she will tell me. Okay, but for now, I only have the percentage there. Okay, so this is the percentage versus the grades, and then if you want to know your GPA, that's how you can take a look as well. Okay, see you in a bit, guys. All right, let's keep going with the lesson. The next little part of the lecture is about cellular energy. We already know the mitochondria is responsible for producing energy, and I told you the process of creating energy involves taking glucose, reacting it with oxygen to produce carbon dioxide, water, and energy. So let's just elaborate on that a little bit, okay? When we say metabolism, we are referring to all the reactions, all the chemical reactions that are going on within your cell, and we can actually separate the metabolism into 2 different kinds. Okay, we have catabolism and we have anabolism. So let's fill this in together. Let me just copy it to the note here. There we go. Okay. So on the left side here, we have a bunch of macromolecules, which are, proteins, lipids, carbohydrates, so on and so forth, and we consume these things from our food. So macro molecules. from food. Okay, so example, carbohydrates. We know this already. Carbohydrates, proteins, lipids. Let me zoom in a little bit for you. And these molecules are really big, and we cannot harness the energy from them unless we break it down. So when you break them down into individual building blocks, right? These are monomers, right? Many building blocks, such as maybe glucose, amino acid, right? Fatty acid, so on and so forth. Right? Then we can absorb them. We can burn them for fuel, and we can use the building blocks to make things that we need. Okay, let's focus on the left side here first. When we take the macromolecules and we break it down into smaller pieces, we use the hydrolysis reaction, right? But there are different kinds of hydrolysis reaction depending on what you need to break down. So generally speaking, when we are breaking things down in the body, we call this catabolism. Okay, catabolism or catabolic reaction. Basically, we break down molecules. Now, in the process of breaking these molecules down, energy will be released. Energy will be released, and you try to capture some of that energy. I'll tell you how to do that later, but energy will be released, okay? And we can store that energy, and then we can use it for different things. At the same time, when you are breaking down the food, breaking down the macromolecule, not all the energy can be captured. Okay? And in fact, a good chunk of the energy is lost as heat. So that's not a bad thing necessarily for us because we are warm-blooded animal. We need to maintain a body temperature 37 degrees Celsius. So after we eat a meal, we feel warmer. It's because of the heat that is being generated when we break down those food. Okay. So if you think about like a light bulb, you don't have to draw this down. I'm just trying to help you understand, put it into context for you. If you think about the really old kind of light bulb, the incandescent light bulb, these things are only about 10% efficient, right? Meaning 90% of the energy is going to be wasted as heat. That's why you touch one of these things, it burns your hand, right? And then the technology got a little bit better, right? Remember, you probably still have some of these at home. These spiral ones. Anybody know what they're called? Right, these are incandescent. What do we call these ones? Maybe my drawing is not too good, but like it looks like a bunch of spirals. Anybody know what I'm talking about? No, yes, maybe. Yeah, these are, yeah, go ahead.
Tungsten or not?
Oh yeah, the tungsten is the original one. Okay, yeah, this one is the fluorescent light bulb, guys, right? Fluorescent. That is about 85% efficient. Okay, and then nowadays most people have like LED, right? And they are more than 90% efficient. Okay, meaning it's completely opposite to the traditional light bulb. Okay, over here they barely generate any heat, right? Okay, so what do you think we are like in terms of the human body? Do you think we are like what percentage do you think um, would be our efficiency? Like, are we like the light bulb, traditional incandescent light bulb, or are we like the LEDs? How efficient is our body in converting food into energy? Hey, Alina said 90%. Anyone else? Just throw me some numbers. Okay, yeah. You guys are very, very optimistic about our body. Unfortunately, we are only about 25% efficient, okay? So 75% of the energy that we get from our food are actually lost as heat, okay? Again, that's not necessarily a bad thing. It helps us maintain our core body temperature, but we're just not very good, very efficient at converting the food into energy, okay? So maybe that's why we have to eat like a couple times a day, right? To keep going, okay? So that's catabolism. On the other hand, over here, once we get these building blocks, we can use them to build things that we need in the body, right? So you might eat some meat to get your proteins. You might eat some cheese. You might eat some beans to get your proteins. But we take those building blocks and we build proteins that we need in our body, right? So to grow our nails, we need more proteins. To make more muscle, we need proteins, right? So all these building reactions, we call it anabolism. Anabolism. Right? So this is to build molecules. And that requires energy. Right? And so the energy we get from breaking down the food is actually used to allow us to build molecules that we need. And so in the end over here, these would be macromolecules needed in the body. Okay, that's all. So collectively, catabolism and anabolism, they are both metabolism, okay? But one is breaking down, one is building, basically. One of the major catabolic reactions that we need in our body is cellular respiration. I already went through cellular respiration with you a couple times already. I can write out the equation for you one more time. It is glucose plus oxygen, and then it's going to produce carbon dioxide plus water and energy. Right? So we can use this energy for building molecules, for moving body parts, and so on and so forth. Part of this reaction is going to happen in the mitochondria, and we actually will spend the entire lecture 5 talking about cellular respiration. Okay, I want to shift our focus to learning about this molecule called ATP. Let me see if I have a picture of that. No, I don't. So ATP stands for adenosine triphosphate. So it's made up of 3 parts. There is the adenine. It's the same adenine that you will find in the DNA base, connected to a ribose sugar, like the one in RNA nucleotides, and it's connected to 3-phosphate. That's why it's triphosphate. Like a tricycle has 3 wheels, triangle has 3 sides. Triphosphate means you have 3 phosphates attached to it. What's so special about this molecule is when you chop off the phosphate at the end, energy will be released. And you can use this energy to do various things. But once you chop off the P, then it becomes ADP, adenosine diphosphate. Okay, so you can think of the ATP, the triphosphate, as fully charged, like your phone, fully charged. After you plug it in overnight, it's fully charged. And then you use it, then now this is depleted. Okay, it has no more energy. So what do? Well, you can charge it up again. You can supply some energy, and that will allow you to put the phosphate back on, and then it's going to be ready for use again. So basically the ATP and the ADP just cycles back and forth in your cell And whenever you need energy, you just break off a P and then you use it. When you are producing excess energy, you can store it for later use. Okay, so a charged battery versus a drained battery. Right. So just think of the pen here, right? Like if you need the energy, you break off the P, and then now it's used up. So you can charge it up again, but then you gotta cap it. You gotta put the P back on, and then now it's good to go again. All right. Let's try to put this together with the catabolism and the metabolism to help you understand a little bit more. So we have the food here. We have the building blocks, if you would. So basically, this is going to come out here and is catabolism, just like before. But then it's going to release energy, right? Release energy and heat, but we're going to focus on the energy right now. So you might not use the energy right away. You want to capture that energy so you can store it for later use. And that is when you're going to take the drained battery, the ADP, you're going to find a phosphate that's floating around, and you're going to pick up the energy, going to pick it up, store it, cap it, and it's going to become ATP. So let's say you have a bunch of building blocks and you want to go and build molecules now. Okay. Maybe I shouldn't put food here, okay? I'm just going to put like macromolecules because you're not really building food, right? Sorry about that. So take the building blocks and you want to build bigger things in the body. That would be anabolism. But that requires energy. So we're going to take our stored energy, the ATP, we're going to break off the P, and then it's going to go back to the drained form, the ADP, and then the P that you've broken off. And at the same time, you're going to release the energy. And that's the energy that can be used for building things. All right. There's a question. This is the process of charging the energy inside the cell. I would say like capturing the energy. Okay? So the ADP is going to capture the energy, and then it's going to store it in the form of ATP. So at any given time, your body is creating new ATPs, and you're also using up ATPs. Right? So these things just kind of cycles back and forth in your cell all day long. Any other question about the relationship between catabolism, metabolism, anabolism, and then the ATP? All right, what do we use energy for other than building things? Well, you can use it for mechanical work, which basically is movements. So if a cell has flagellum that it needs to beat, that's going to need ATP. If you move your muscle, that's going to need ATP, right? Beating of the heart, breathing, thinking, all those are mechanical work. It requires ATP. Chemical work, we already learned about enzyme. So enzyme lowers the energy requirement for reaction, right? But nothing is free. Something has to make up for that difference, right? Like the analogy for the scholarship, you don't have to pay as much, but someone has to make up for the difference, right? Like maybe from the scholarship, like someone has to donate to that scholarship, right? So in a sense, that's what the ATP is for. The ATP is going to make up for that difference in energy and allow the reaction to take place. Transport as well. Okay, as we will learn probably, I don't know, maybe next class. We don't have a lot of time. If you drop something, I just let go and then it will drop, right? But to pick something up, then it requires energy. Maybe not so much energy for the pen, but think of like a 10-pound dumbbell. You just let go, the dumbbell will drop on its own. But to pick it back up and lift it up, like a 10-pound, 20-pound dumbbell, that's going to take energy, right? And in that regards, it's the same for transport. If you try to move things from high concentration to low concentration, then it's just going to happen on its own. But if you try to move things the other way around, then that's going to require ATP. And the rest of the class will be the rest of the lecture actually will be talking about the different kinds of transport, what kind needs ATP and what kind does not. But basically, ATPs are used for various things, and these are 3 common ways of using it. Okay, you cannot live without ATP. Okay? And to make ATP, you need to constantly do this reaction. And to do this reaction, you need 2 things. You need food. Okay, that's why we have to eat. And we need oxygen. That's why we have to breathe. Okay? And we're going to learn a lot about this process in lecture number 5. Okay. I am just going to do this one with you and so that we can wrap up the lesson. Catabolic reaction. Is this building or is this breaking? I just want to see if you...
Breaking.
That's right. Yeah. Then if you have trouble remembering it, you know, catastrophe, right? The word catastrophe, like an earthquake or something, tornado. In a catastrophe, everything is destroyed. Everything is broken down. So kata is the same prefix here. Catabolic reaction, breaking things down. So what happens when you try to break things down? It consumes more energy than they produce. That's not true. Because if you look at the notes we wrote, catabolic reaction actually release energy that we can use. So it releases more than it absorbs. Breaks down polymers into monomers. That's correct. Produce excess heat to warm the body. Yes, because we are only 25% efficient. Only occurs in human cells and not other cells. Nope, humans are not that special. Other organisms will be able to do similar processes to help produce energy for their needs, right? So the correct answers are B and C. Okay. Let's take a look at this. I don't think we have a lot of time. I think we can probably only do 5 or 6 more slides and then we will have to call it a day. Because this stuff over here, we have to take a note that is gonna last at least like 15-20 minutes. So we won't be able to do that today. Anyways, the next part we're gonna do is a type of transport. These are red cells that you're looking at, and I'll tell you next class why they all look, some of them look kind of weird. Transport. Transport is the movement of things across the cell membrane. That's our focus, right? Remember, the cell membrane is selectively permeable. It lets some things go through and then it blocks other things, harmful things which is going to stay out, resources, useful nutrients, it's going to come into the cell, right? So we're going to look at the different ways of moving things, transporting things across the cell membrane. Starting with diffusion. Okay, diffusion. Diffusion, I'll just read the definition to you first. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. If you add a few drops of food coloring to a flask of water, the dye is going to spread out on its own. You don't even have to swirl it. Sure, you can swirl it to make it go faster, but if you just leave it alone, the ink is going to spread out on its own. So in the center here, that's where it is high concentration. There's a lot of it. And over here, this is low concentration. So on its own, spontaneously, it will go from high to low concentration. No energy is required. Things will naturally spread out like that. Okay. If we are in a classroom, I can take a Febreze, you know, those air freshener, and I spray it at the front, right? Psst, like that. And then after 5 minutes or something, you'll be able to smell it at the back of the classroom because it goes from high concentration where I sprayed it and naturally spreads to throughout the room to places with low concentration. Okay? That's called diffusion. Goes from high to low zero energy requirement. It's gonna just happen on its own. Got it? Now, there are different factors that will affect the rate of diffusion. There are different things that would speed this process up or slow it down. And we're going to look at 4 of these factors. Number 1, the size of the molecule. Right? It's not too difficult to understand. If it's small, it's going to be able to move faster compared to a bigger one. Okay, if it's really big, it takes more energy to move it, right? So if the molecule is small, like over here, oxygen, it's going to be able to diffuse out faster. If it's big like this one is a glucose molecule. You can see it has more atoms in it. So it's going to have, it takes more energy for them to spread out. They will still spread, okay? It's just going to take longer. There is comment. Yep, exactly. It says, that's why an essential oil diffuser is called a diffuser. Yeah, exactly the point. Okay, it diffuses the scent out to the whole area. Okay, factor number 2, it's temperature. Right? If it's hot, then it tends to spread out faster. Right? Think about like I don't know, this on a hot summer day. Okay. Every Wednesday is garbage collection for us, okay, for where I live. And on a hot summer day, like the streets smell so bad because all the garbage is on the sidewalk, and then the temperature is really hot, so all the scent is like coming out, right? And you can smell it in the air. It's diffusing out, right? Today is Wednesday, also garbage collection day, but it's so cold outside. I just went shovel the snow before I come to class. I can hardly smell anything, right? The cold temperature keeps the diffusion rate very, very low. Okay, so the molecule is not going to be going all over the place. Okay, so size matters, temperature matters when it comes to how fast these things are going to be spreading out. The next one is called the steepness of concentration gradient. Okay, in the picture at the top, which way are the red balls going to go? Look, there are a lot of red balls over here, and then there are none. Right? So which way will the red ball diffuse? To the right or to the left?
Right.
That's right. It's going to go to the right from high concentration to low concentration, right? So over here, there's a lot of them. I'm going to say it's 100%. And over here, there are none. So I'm going to say 0%. So the difference here, the difference is 100. Okay? If we take a look at the bottom picture, it is still high concentration on the left and low on the right. But this time, instead of 100%, I'm going to say maybe 70%. And over here, there are some to begin with, so maybe 20%. So the difference is only 50. It's not as great as before. So diffusion will still happen, but it's not going to happen as quickly as the top example. And that's what we mean by the steepness of the concentration. You can think of dropping a ball down the ramp. If the ramp is steeper compared to like a less steep ramp, and you have a ball and you let go, this is gonna go down faster, right? And this is still gonna go down, but it's gonna take some more time, right? So same concept here. The greater the difference between two locations, the steeper the difference in concentration, the quicker the diffusion will happen. This one, it will still happen, just not as quickly. Okay? Size, temperature, steepness of concentration gradient, and then the last one is the charge. Okay? If things are charged, if the molecules that are doing the diffusion is a charged molecule, then it's going to have an effect on the rate of diffusion. Over here, you see there's a bunch of H+, and positives do not like each other. They like to stay away from each other as far as possible. So if you have a bunch of positive here, they're going to push each other away like that. Right? So initially, it's going to cause them to spread out really, really quickly. But as they spread out, like if over here there's too much positive charge gathering, then they might push some of them back, right? So eventually they're going to reach some kind of equilibrium, we call it, some kind of balance. But initially, it will cause them to spread out very quickly if they are all having the same charge. All right. Okay, I'm going to take a last note with you before I let you go. This thing is confusing for a lot of people, so please ask questions if you don't understand, and you know, ask for clarification if you need to. So I'm gonna draw. Okay, this is called osmosis. Okay, osmosis is diffusion of water across a membrane. Okay, so I'm gonna draw a tube here, and it's gonna be a U-shaped tube. Okay, just try to copy what I have. Do right here. There we go. Okay, so we have a U-shaped tube. Now, at the bottom of the tube, and this is very important, we're going to put a membrane. And what's important about this membrane is this membrane only allows water to pass. Okay, nothing else gets through, just water. Okay, the little gaps here are very, very fine, very, very small. Only water can pass through. Then I'm gonna fill this thing up with water. Okay, I'm gonna put some water in here. You can fill up yours too. And there we go. And we're gonna fill up the other side as well to the same level. Okay. Then I'm gonna dissolve some solute in it. Okay, I'm gonna dissolve some, doesn't matter what it is, some salt maybe. Okay, and then I'm gonna put them in here. Oops, that's a little bit too big. Okay, I'm gonna put them here. Okay, and I'm gonna put a few here. There we go. Now, you don't have to answer me. You can just think about it in your head when I tell you the questions. Okay, number one, can the red dots move from one side of the tube to the other? Can the red dots move from one side to the other? Yes or no?
No.
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 That's right, because I told you it cannot, right? I put a gate there that specifically block these things. Only water can pass through. Okay? Keep this in mind as I continue with the note, okay? The red dots cannot cross. Okay? These are solute. They cannot cross the membrane. Okay? I forgot to label, but you know, this is the membrane. All right, which side has high concentration? Let me do a poll and see if you guys know. Okay, which side has high concentration? Is it A, the left side, or is it B, the right side? Which one has higher concentration? We're gonna taste it. Which side is gonna taste saltier? Right, if the red dots are salt, which side will be saltier? Okay, good, good, good. Most people got it right. The left side is saltier. It has more salt, right? And here is the $1 million questions that people have trouble with on the test. All right, I'm going to relaunch the poll. The question is, if I leave this thing alone, which way will the water move? Water can move, right? It can go through the membrane. Is it A towards the right? Is it B towards the left? Or C does not move? Okay. I'm asking about the water. Which way will the water move? Okay, not too bad. Anyone else want to try before I close the poll? All right, thank you. The correct answer is B. Right? B. Water will move to the left side, right? To try to balance out the concentration, right? So if the membrane only allows water to move, water moves to the left. Okay? As water moves to the left, concentration on the left will decrease because now you're adding more water to it, right? You're going to dilute this while the concentration on the right will increase because you're removing water from it. You're reducing it, right? So this continues. This will continue until the concentration on both sides,
Equal.
that's right, are equal to each other. Okay? So the water level will rise over here and the water level will drop over here. Right? Now you can try to visualize this a little bit. If it's very abstract for you, you can imagine this, and if imagining it doesn't work, you can physically try it. Okay, although I don't recommend it. You can take a small spoon of salt. Okay? Try to visualize this in your head. Take a small spoon of salt, and then you put it in your mouth. Okay, don't swallow. Just put it in your mouth and hold it for 30 seconds. Right? In this hypothetical situation, can you visualize what's going to happen to your mouth? Spoonful of salt in your mouth, hold it, don't swallow. What's going to happen?
Water comes out in mouth.
That's right. You're going to, start drooling, right? You're going to have like saliva coming out, right? That is exactly what this situation is like. If the salt cannot go into your cells, yeah, they're too big, right? But it will draw water out of your cells. It will draw water out of your mouth, basically, right? So that's osmosis, right? When the solutes cannot cross, water will always move towards the side that is saltier, so to speak. Okay? Water will always be attracted to the side that has higher concentration, and that is osmosis. Okay? So when people try to preserve fish before refrigeration, right? They try to preserve meat, they add a lot of salt because salting it basically sucks the moisture out of the food, and with no moisture, bacteria cannot grow, right? And that's a great way of preserving food. It's not very healthy though. Okay, all those extra sodium is going to cause all kind of health problem, but better than starving, right? In the past. And that's it. Okay, so here is you can see it's another example. It's reverse. We have more salt on the right side. Water is going to move to the right, and then it will continue to suck water to the right side until the concentrations are the same on both sides. Okay, so that's it for today. Now, before you go, let's just quickly take a look at what's going on next week. This is to remind you as well as to remind me. This is the wrong class, sorry. Yeah, there you go. So next we are here today, and I think we're right on schedule. On Monday, we will do the rest of lecture 4. I will tell you a little bit about the lab, and then I think I can finish lecture 4 on Monday. I am not 100% sure. If I don't, then we're gonna have to have a bit of a class on Wednesday. Otherwise, if we finish lecture 4 on Monday, then no class on Wednesday. You can just focus on working on your lab. I mean, I do have a few minutes here, so I will show you what the lab is and where to get it. When you log on to Luminate, you can scroll down. Let me just close this thing. You can scroll down to Beetroot Lab. That's what you're going to be doing, okay? The Beetroot Lab. You can open the folder, and there it is. Okay, so when you open the Beetroot Lab, there are some description here. You need to read them before you do the lab. And then there will be questions that you have to answer. Okay, so for example, what is the purpose of the lab? What is the name of, so like you're gonna read all these things, okay? And hold on, where is this? Am I missing something here? I'm gonna have to double check. But anyways, you're gonna answer these questions, and it's gonna be done kind of like the online quizzes, right? Some questions will have help in them. Like for example, we ask for the dependent and independent variable. If you don't remember what those are, then you can watch the help video before answering. It's pretty straightforward. And then I did the lab. It's me in the video. All you have to do is watch what I did and then answer questions related to it, right? So like which beaker is the control experiment, control of the experiment. So you have to pick one. And if you forgot what a control is, there's a video that reminds you about it. And basically you go through the whole lab. And the only one that might give you a bit of a problem is like graphing here. Have you guys learned how to graph yet in your computer course?
Yeah, we learned pie chart.
A pie chart, okay. Oh, well, this one you're going to be creating a line graph. If you don't know how to do a line graph, that's okay. I mean, like I have a video for you as well. Okay, so I thought you've done it in computer.
This
but
is bar chart.
line graph, line.
Okay.
Yeah. So anyway, there are lots of help in the video in the lab itself. So I don't think it's going to be too big of a problem. And then that's all you have to do. Okay, just to answer all the questions. It's 10% of your mark. How much time have I don't think I don't think I put a time limit on this thing. Okay, so take your time, but you have to submit it before before the deadline. Okay, and very importantly, you have to answer these things based on the videos. Okay, in the past, what people has done is they try to look the answer up on the internet, and it's not right. Okay, so some of the answers on the internet, it's not correct. And I will know. Like I said, I taught this course for like many years, right? So when I see something that's from the internet, I will recognize it. And a lot of times, people start talking about, you have to use the alcohol for this. But I haven't used any alcohol in the lab, right? So I would know it's not your own answer. So don't do that, and don't use AI to come up with the answer. Again, it's super obvious when I mark it. Just use your own words. Watch the videos. Should be more than enough. Okay. There's a question: will the graphs be submitted and done in the test link, or are we supposed to? So you have to use your own software to make the graphs. No lockdown browser when you're doing the lab, so you will have access to Office. But you don't have to use Office, okay? Over here, this one, the video over here, I actually used a Google Sheet, which is free. Okay, all of you can have a Google account and access Google Sheet, and it's more than capable of generating a line graph. And once you've generated it, you have to attach it to your to the answer here. Okay, so there's an attach button that you can use. Do not email me anything. Okay, don't email me your graph. You email me your graph. I have to find out which quiz you are doing, and it's just like too much confusion. Okay, that's unnecessary. Just attach the graph onto it. You can take a screenshot, right? A lot of you have this snipping tool. If you're using what do you call it? Like Microsoft, if you type in snip, right? Like here, snipping tools, it will allow you to take screenshots of your screen. Okay, it's just taking a long time to load, but there it is. Okay, press the new screenshot, and you literally can copy anything on your screen. So let's say this is your graph, and then you just highlight the graph, and it will be there. You see? Okay, and then you save this, and then you upload it. Okay, it should be very straightforward. You guys have to upload things for your math class, right? Don't you? Am I correct when you do your math quiz?
Yes.
Yeah.
Yes.
Okay. So you are familiar with the uploading process. Thank you. Is
So
this
if I do it in Excel, then attach it, is it okay?
you don't attach the file? Okay. You attach the graph. Right? Attach
Yeah.
the graph. Yeah. Thank you.
Mhm.
This lab is about two things. It's about scientific method, which is lecture 1, and it's about membrane, cell membrane. Okay. There will be an intro video in the lab when you do it. So after you watch that, it will make more sense to you. Okay. So that is it for now. Quiz number 2 will be, later on. It's next week, so we don't have to worry about it at this point. Yeah, that's all. If you have any questions, feel free to email me. Otherwise, enjoy the rest of your week, guys. Take care. Bye bye.